welcome to the Bio 1-2 lecture. We'll be doing a bit of a head start, a little intro today, looking at all things Unit 2, so focusing on what will come up this term and throughout the rest of the semester as well. My name's Loz. I'm going to be taking you through the lecture today. Before we do begin, I'd just like to acknowledge the traditional owners of the lands from which I'm presenting from today, so the Wurundjeri and the Boonwurrung people of the Kulin Nation. And I'd like to pay my respects to their elders, past, present, and emerging. So some of you guys may be familiar with the ATAR Notes lectures. You may have been to a couple already earlier in the week. You may have been to some, earlier in the year. Essentially, we run these lectures. They are for, generally year 11 and 12, sometimes a bit younger. And they are basically high school resources that are run by past students. So again, you may be familiar with ATAR Notes. We have a couple of other resources. I tutor for ATAR Notes, so we have a couple of things that may be helpful for you that you may want to check out. If you find this lecture helpful, if you found some of the others helpful, you can have a look at some of the others that may be running. You can get these recordings and things like that and the slides as well. We also have a lot of other resources, particularly our study notes. You guys may be familiar with the ATAR Notes discussion forum. There's quite a lot of stuff you can have a look at. I know most of you may be in year 11, maybe year 10 as well. So, some of these things like an ATAR calculator, maybe more so for next year, but there's definitely a lot of stuff that can help you if you're doing 3/4s or anything else like that. So feel free to check it out. You can just head to the website and just register and make an account if you are interested. And if you think it might be something that you're wanting to look at. If you do feel like you want a little bit more than those resources that are completely available on the website, we do have a couple of other things that you can check out. So as I was just mentioning, TuteSmart is the tutoring company that's run by ATAR Notes, so where I tutor bio, for example. We also have some other study guides, this little website, this little program called Ed Unlimited. Essentially what Ed Unlimited is again, you may be familiar with some of the ATAR Notes study guides, topic tests, course notes, all those sorts of things. Ed Unlimited essentially is where they are collated all together and they're available. It's almost like a little Netflix, but for study guides. So if you think that you may want to check that out, or you may want to look into some study guides or some topic tests, feel free to head there. There's a lot of bio stuff, a lot of bio 1-2 stuff, bio 3/4 stuff, and a lot of stuff on the other kind of VCE subjects as well. So feel free to have a look if you are interested. Okay, so essentially what we're going to be covering today is, as I was mentioning, a little intro into Unit 2. So there is, as you can see, quite a bit to get through. We're going to be looking at the sort of things surrounding DNA. So we're looking at genetics, we're looking at chromosomes, we're looking at meiosis, mitosis, so things on a very cellular level. And then we're going to end with a little bit of biodiversity stuff that belongs to the second area of study of Unit 2. And then finally, just a couple study tips. If any questions pop up, please feel free to pop them into the live chat. You can also feel free to email me if you have any questions that we don't get to, or any other things that you'd prefer to email. I'm more than happy for you to, send me anything that you want me to look at. In terms of me and my experience in bio, so I did bio 1-2 when I was in year 11, so I went to bio 3/4 when I was in year 12. I graduated in 2020. I got a score of 45 in bio, and at the moment I'm studying in my third year of med at Monash. If you have any questions about, bio 3/4 or anything about uni as well, you can feel free to pop that in the chat, or again, feel free to email me. But yeah, I did bio 1-2 again, as I mentioned, in year 11. It was, I think it's quite a good foundation for bio 3/4. There isn't lots of people do bio 3/4 without doing bio 1-2, but I know that when I went into bio 3/4, I felt happy, and I felt a little bit more confident, I suppose, knowing that I had kind of a year to practice ultimately answering bio questions. I'm sure as you have figured out, from the first 6 months of this year, and I'm sure as you'll figure out if you continue on bio 3/4 next year, there is a very specific way to answer bio questions. I would say it's more specific than some of the other sciences because it is a lot more wordy. There's not really many formulas when you think about bio and things like that. So there's a really specific way to kind of use terminology, use your definitions, and answer questions in a certain way to ensure that you're getting full marks. And so I think that's sometimes why bio can be seen as a little bit tricky, because there's obviously a lot of content and a lot of complex content. Um, but it's almost another part of it is also learning how to respond to questions, how to answer things in the correct way. And I think that's what Bio 1-2 is really, really good for. So hopefully these first 6 months of the year, you've kind of had a little taste of that. And going into the next 6 months, I think it's a really, really good time, again, to use this as almost like a bit of a draft year for 3-4. So essentially figuring out, where your kind of common mistakes are, what you're struggling with in terms of bio, not so much in content, but in general, is it grasping things in class, or is it, are you struggling with extended response questions, or maybe it's your multiple choice. So having a little kind of think about those things is really good to figure out in year 11, or you guys maybe in year 10, to figure out in 1-2. So then by the time you get to 3-4, you're really confident with your ability to take bio, assessments, bio tests, that sort of thing. Okay, hopefully that makes sense. Again, any questions, just pop them in the chat. But this is what we are running through. It's very much all to do with sort of DNA and then a little bit on biodiversity ultimately. Okay, so in terms of what DNA is, I'll say this as well at the start. Bio is a really almost definition-based subject, with a lot of your short answer questions in particular, some multiple choice as well. You need to know your definitions. I remember my teacher really, really saying this and emphasizing this a lot when I was in both 1-2 and 3-4. Keeping a glossary is really, really important. It's not too late to start one now. I know you may have just had your exams. You may have another exam, at the end for Unit 2. Some schools do it differently. They might do, you might have a Unit 1 and 2 exam at the end of the year, or you may have your mid-year exam and your end-of-year exam. But going into Unit 2, I would really suggest, if you're not already, to keep a good glossary, and just constantly kind of chip away at it after every class, just add some terms to it, because I think that's the best way to do it, and that kind of lets it sit in your head, as opposed to like a week before an exam trying to really smash out a glossary. It might not be as beneficial to you. So I really, really highly suggest. And then again, keeping that sort of skill, or I guess that strategy, when you're going into 3-4 is also really, really important, because again, there's just more terminology. The questions are still definition based. So that's kind of a tip that I would suggest. So in terms of what DNA is, I'm sure you guys will be familiar. You may have looked at it in junior science. So DNA is essentially our source of genetic information, basically our instructions telling the cell what protein to make, what to do ultimately. And then obviously this is passed down from individual to offspring and so on. Okay, so in terms of our genes, so DNA is the NA stands for nucleic acid. So that's our kind of really long, big molecule, sort of what we're seeing here. A gene is a really small section of that DNA that codes for a specific protein. So we can see that it may, determine characteristics. It may be for something that is within the cell, maybe for something that is external to the cell. Again, I think I did say it codes for a protein, but you have different genes that do different things. So it may be a protein, it may be, a hormone, it may be an enzyme, or it may be for regulation. Genes sometimes may not even code for anything really that meaningful. So some, sections of genes, some sections of DNA may not be for anything either. So it's really almost case by case when you look at different genes. But it's important to understand that you've got your huge, big, chromosome, your huge nucleic acid, and then a gene is just a little small section of that. Okay, so then zooming in even more, so looking at our genes, looking at our specific nucleotides. So your nucleic acid is what we would call a polymer. So it's a really big molecule, and poly meaning many. It's made up of many small structures, and these small structures are called monomers. So mono meaning one. So your nucleic acid, your DNA, is your polymer. Your nucleotide is your monomer. So it's just that one little unit. You can think of like, bricks making up a house, or like Lego blocks or something like that. But it's important to kind of understand that idea. It's something that kind of recurs in bio. So we have our nucleotide. They are seen here. This picture, obviously, it's a bit of a combination. But we have our nitrogenous bases running here down the middle. We can also see them depicted here. And then this little double helix is what you can kind of call your DNA backbone. And so it's termed a sugar and phosphate backbone. So here we have our phosphate group and then a sugar, and then a phosphate and then a sugar, phosphate, sugar. And you can see that that just runs down there. I like to think of it as like a ladder. So if you think of like your sugar and phosphate making the sides of the ladder, and then the nitrogenous bases make those horizontal rungs. That's how I kind of like to think of it in my head. And so one nucleotide is just one of these little things. So just one phosphate group attached to one sugar attached to one nitrogenous base. So those three components, that's what makes up your nucleotide. And then obviously you put a bunch of them together. That's when you've got, if you can think about like one strand of DNA, and then you obviously pair it with another strand, and that's when you get your full molecule. So in terms of our nitrogenous pairing, we've got this idea of complementary base pairing. So complementary is also something that comes up in bio a lot. It's this idea that adenine and thymine and cytosine and guanine, they always go together. So they are complementary, they match each other, and that's how they'll always pair. So it happens spontaneously, you don't need to force them together. It's this idea that adenine and thymine will always pair up together. So this is a really, really important thing to remember, because you'll often get questions about it. You might be given like a single DNA strand and then asked, what would be the corresponding one, just matching your A's and your T's and your C's and your G's. But that's a really, really important thing to understand. So again, a double-stranded helix. So we've got our two strands that run anti-parallel. And then we've got the helix part, which is just this idea of it being kind of like a corkscrew coiled around. So hopefully that makes sense. Okay, and then again, back to kind of zooming out to our genes. So this is our little double helix here. So a nice long string of these nucleotides is what makes up a gene. And again, so if we're thinking about a protein, for example, that's kind of the most common example that we use. So you have a gene and it will be transcribed. So this process of transcription, and then it turns into this molecule mRNA and it'll become a protein. This kind of detailed process is what you look at next year in Bio 3-4. You don't have a look at transcription this year, but it's important to kind of bear that in mind, that that's something that you look at, but also to understand for this year that you have your gene, goes through transcription, goes through translation, gets to this protein, and then that protein is expressed and it does what it needs to do in the cell. So that's the important connection that you have to make in terms of genes, and then that gene being expressed and it resulting in, if it's a protein or if it's anything else, and then that having its role in the cell. So hopefully that makes sense. We can see here. So the length of the gene, the specific base sequence determines what comes out. So your length of the gene obviously determines what gets expressed, and the order of those nucleotides is also really important, because, we've talked about seeing our little rungs here. So this order of nucleotides is what will determine basically what protein gets made. So it's very, very specific. So, we'll talk about mutations a little bit later. But if you had a mutation in one of these and your adenine got switched to a cytosine or got switched to a guanine or something like that, depending on where that mutation is, that may have, a big impact on what's actually produced. It may create a faulty protein, for example. Okay, so looking at alleles. So alleles are ultimately, we've got our little genes, and so our genes, code for certain proteins, code for whatever. Ultimately, that results in a certain characteristic. So you've got a gene for a certain protein, and then, thinking about consequences of that, maybe that gives you a certain hair color, a certain eye color, a certain height, for example. So alleles are basically variety between these specific characteristics. So you have got, this little gene that, let's say, codes for hair color. So an allele will be, codes for brown hair, codes for black hair, codes for blonde hair, for example. So they're all the same gene. It's the exact same gene for hair color. It's found in the same place. It's just that individuals have different variations of this same gene. So hopefully that makes sense. And you can see that it's found on the same place. So if you saw, if I have black hair, if my friend has blonde hair, if we find the specific chromosome, the gene for hair color will be found in the exact same place. It'll just be one codes for black hair, one codes for blonde hair. So the genome is ultimately the collection of all of your genes, all of your genetic information. And it's basically the base pairs written out. So it's got all of your bases, the sequence of all of the bases ultimately in all of your DNA spelled out. So that's your genome. So you can see it's a very, very, very big thing. We've got 134 million base pairs, but it's ultimately just a sequence of what's going on. And you can kind of think of like, I feel like teachers always use this analogy of like recipe books and, instructions, all that sort of thing. So if your genes are the recipes to create, the food that is your hair color or whatever, the genome is like the big recipe book or like the series of recipe books or something like that. So it's just a big collation that contains all of the genetic information ultimately. So the idea of the genome is that it was a product of this thing called the Human Genome Project. So it was this big thing, about sequencing the human genome. So it was really helpful because obviously, you might think like, oh, what's the point of this? Or like, cool, you've got a lot of information. But it's really helpful for a bunch of things. So it was a very big project, but it obviously was very helpful in the kind of setting of disease, particularly genetic diseases, thinking about what genes code for what proteins, what genes may even interact with each other. There is another, so the genome is your genes. Something else is your proteome. So these are your proteins, kind of the equivalent of a genome, but for proteins. And these ultimately, the genome can kind of help in looking at the proteome and thinking about that and the importance of the proteome, thinking about having this collection of what proteins you express, that's also really, really important. So having this kind of collection, this kind of sequence is a really important, it's not really a discovery, but an important project that happened. And you can see that this is just only like 3 of the benefits. So you can sequence genomes of other organisms, this idea of determining relatedness between species. This is something you'll look at in year 12. And then understanding the role of genes as well. So talking about the role of genes, for example, in certain diseases, in the progression of disease, things like that. But again, there's heaps more benefits that sort of came from that. So looking at chromosomes, so we've talked about, you've got your little double helix. Your chromosome is basically the condensed sort of storage device, I guess, of your DNA. So we have your DNA is like, you can kind of think of it as string almost. And it's just coiled and condensed very tightly, and then it makes up this bigger structure that is a chromosome. So I think that's something that might confuse people. Sometimes you might think that the double helix itself is in like the shape of a chromosome. It's not like that. The double helix is like a piece of string. So if you imagine a string and it's just coiled, and then that string is then, wrapped around these proteins and then condensed into a really big molecule or big thing, that's your chromosome. So try not to think about it as like your DNA is in the shape of a chromosome, because that isn't the right way to think about it. So in eukaryotes versus prokaryotes, so eukaryotes are your more complex organisms and your prokaryotes are your simple, often unicellular things. So like your bacteria, for example, is a prokaryote. So eukaryotes, like, for example, animal cells, we've got multiple chromosomes and they are linear. So they're kind of like this shape or just a single line. So those are the chromosomes that we kind of think about for us. Whereas in a bacteria, they have one chromosome and it's in the shape of a circle. So that's kind of a difference to remember as well. So in terms of your chromosomes, I was talking about how DNA is like string wrapped around a protein. So these proteins are called histones. They're like little balls, I guess. And yeah, I like to think of it as like if you have a tennis ball, which is your histone, and then you've got, string or yarn or whatever, and you're just wrapping it around. That's how I kind of like to think of it. So then you basically chuck heaps of histones together, and then they will, condense, and they're super, super duper squashed. and then you basically call it chromatin. So some of the terminology is a little bit hard to comprehend, but it's just important to understand how DNA is really condensed, and you can imagine how much DNA there is in a cell. So you've got your little histones, they're kind of chucked together, they're coiled and condensed even further to create this chromatin. This chromatin is then again folded, and basically you've just got more folding, more condensing, and then that gives you your chromosome. So I think we've got a little picture, yeah, coming up here. Okay, this should make a lot more sense. I feel like I've been talking and it might be hard to comprehend it, but you can see so here you've got your base pairing. This is, your nucleotide. This is the smallest level we're thinking of here. You've got your double-stranded helix, and you can see that this is like a little string, and this curls around a histone. So you can see you've got 8 histones, think of 8 tennis balls sort of coming together. And then you've got around each of them, you've got this string kind of wrapped around. And then these create their own little balls. So these are your like little nucleosomes. And then those curl around even further. And then you can see that those squish around even more to form a chromosome. So hopefully this picture puts into context sort of how much DNA you've got going on, all of that genetic information, and how chromosomes are very advantageous because they basically pack all of that into a really nice tight spot where it just sits in the nucleus. When you're thinking about a prokaryote, we talked about how it's a single circular chromosome, so they don't, have histones or anything like that. And that is fine because it can just be chucked in the middle of the cell because they're not very complex. There's not that much to them. Whereas if we were to do that, just to chuck all our DNA, kind of, shove it together and put it in the cell, there would just be way too much and it would take up all space and it would be horrible. So it's this idea that it's just extremely tightly condensed and then put into the nucleus. And so you can just kind of tuck away that genetic information. Okay, so our gene loci, again, we kind of talked about alleles. So loci is basically just a way of describing where a gene is found on a chromosome. So you will use, it might be on the P arm or the Q arm, this idea if it's close to the centromere, stuff like that. But then they'll have specific numbers and that will tell you exactly where on the chromosome this specific gene can be found. So locus is a word you might see. It's a gene locus. It's just where the gene is found. And then to locus is a loci. Okay, so here is terminology you definitely have to know. So this is what your chromosome will usually look like. I know that we're all kind of used to seeing it in this picture, but it looks like this. And then when you replicate DNA to get ready for mitosis or for division, that's when you get this sort of typical X picture. So here we have homologous chromosomes. So homologous chromosomes are basically, they're the same chromosome. They've got the same genetic information, but they've come from separate parents ultimately. So that's why we have, when we think of chromosomes, humans have 46 chromosomes, but 23 pairs, because you get 23 from one parent, 23 from the other parent, and then you get 46. But you can see how you've got chromosome 1, 2, 3, 4, 5, 6, all the way to 23. And so you've kind of got like 2 copies of 1, 2 copies of 2, but then they'll obviously be slightly different because they're from different people. So hopefully that makes sense in terms of homologous chromosomes, why we call them homologous, because they're the same but different. Same but different is kind of what we're looking at. Centromere is the center of the chromosome. And sister chromatids is when you duplicate or replicate your chromosome, and then you create this little X shape, sister chromatids. It's just the 2 ends, and they'll always be joined by the centromere. Okay, so you have different types of chromosomes. You've got your sex chromosomes and your autosomes. So again, when we were talking about humans, we've got 46 chromosomes. So we've got 23 pairs. We've got 22 pairs of autosomes. So that means 44 autosomes. And then 2 of them are sex chromosomes. And so these will determine if you are essentially female assigned at birth or male assigned at birth. And so you will get your 2 X chromosomes for female and then an X and a Y for male. So these are your sex chromosomes, and that's what determines one's sex at birth. And then autosomes are just kind of all the other chromosomes that aren't sex chromosomes, really. Yeah, so hopefully that makes sense. This is something to be aware of. I feel like some schools may emphasize it more than others. It's not really a major thing, but I do remember kind of reading about it in textbooks and stuff like that. Um, but it's just how some chromosomes have their centromeres in different spots. So usually we're used to seeing chromosomes, depicted in the middle. So this idea of metacentric, so there's equal space between, between the centromere and the end of the short arm and the end of the long arm. And then the centromere might be closer to the top or kind of like halfway to the top. And there's just different ways of describing that. It's not a super important thing, but again, it may come up in your SAC or assessment, it may not. But yeah, it's just something to be aware of, that you can have the centromere depicted at different spots. And when we look at a karyotype, you can see the chromosomes, you can see that they look a bit shorter. Or like, one arm is obviously shorter than the other, or one arm is a little bit more equal. Okay, so your chromosome number differs based on your species. So we've got 46 in us, but other organisms will have a different number. So for example, fly has 8 chromosomes. And again, you can think about how complex organisms are. Sometimes, if you're more complex, you might have more chromosomes. So hopefully that makes sense. In terms of ploidy, so you hear the terms haploid and diploid. We'll talk about polyploidy a little bit later. But haploid basically means half of your normal number of chromosomes. So you've got one set of chromosomes. So like one of each of the 23 pairs. And then diploid, so di, kind of meaning two. You've just got that kind of full set. So 2 sets of chromosomes. So and we use N and 2N because, again, in different organisms, you have different numbers. So in humans, 2N equals 46, N equals 23, whereas in the fruit fly, 2N equals 8, N equals 4. So that's why you see N and 2N, because it differs between species. But in humans, you can just think about haploid being 23, diploid being 46. So karyotype is basically when we are able to visualize the chromosomes found in a certain organism. So we can have a look here, 2 human karyotypes, which is from a male and which is from a female. So remember we talked about your autosome, so that's pairs 1 through 22, and then your sex chromosomes are usually written down there or shown down there at the bottom. So you can see the karyotype on the left has got 2 X chromosomes. The karyotype on the right has 1 X and 1 Y. So the one on the left would be female, the one on the right would be male. Hopefully that's making sense. And you can see they're ordered, as it says here, based on length or, size and the centromere position. So you can see that number 1, 2, 3, they're quite long. And then as we get to the ones at the end, they're all a little bit shorter. And you can see kind of the centromere being up here or up here. But yeah, this is what your typical kind of karyotype will look like. It's important, we'll get to chromosomal abnormalities a little bit later, but understanding what a normal karyotype will look like. So then you can basically spot out if there's polyploidy, for example, or if there's a missing chromosome or an extra chromosome, for example. Okay, so hopefully this makes it a little bit clearer as to what they look like, but you can just order them and then they will be stained. So you can see here, again, the stain is a little bit hard to see in here, but they have banding, and this banding will kind of signify different gene loci and will tell you where certain genes are. So that is, again, really helpful from a karyotype. So they are useful in also noting abnormalities within the chromosome itself, such as banding patterns, or abnormalities in the number of chromosomes. So karyotypes may be done at birth if a chromosomal abnormality is suspected, for example. Okay, so why might karyotypes be carried out when the cell is in metaphase? So I know that we're getting to mitosis a little bit later, but if you recall, we talked about how chromosomes will look like that little X, so they'll look pretty big when they've been duplicated. So the idea is that when you are not preparing for mitosis, so ultimately when the chromosomes aren't being replicated, they're just kind of hanging around like loose in the nucleus. Well, not loose, but they're a bit, thinner. They're not as condensed. Whereas when you're getting ready for mitosis, you're obviously replicating this DNA, and it's becoming a lot more condensed. And that's when they're most easy to visualize. That's when you can see them. So when we get to mitosis, you can see mitosis through a microscope, and you can see the actual chromosomes visibly through a microscope during mitosis. But when a cell is not dividing, you can't really make out chromosomes because of that idea that they're not condensed. So that's the idea there. Okay, so an example of a structure found in DNA, select the option that best represents the figure. So the idea that we're looking at here is we're looking at our histones forming our nucleosome. So we have our histone here. Remember that we've got 1, 2, 3, 4, 5, 6, 7, 8. So we've got 8 little histones that are coming together. And then once they have the DNA spun around them, it's titled a nucleosome. Okay, so we're looking here at heterozygous and homozygous. Again, very important terms to understand. This is when we're getting into the discussion on alleles. So heterozygous basically means that you have got 2 different alleles, and homozygous means you've got 2 of the same. So this can be homozygous dominant, it can be homozygous recessive, but homozygous just means you've got 2 of the same pair of alleles. And so we can see this here. So this is 2 dominant alleles, 2 recessive alleles, and then if you've got 1 of each, then that would be heterozygous. So that's kind of what we're talking about there. Homozygous recessive if you've got 2 recessive alleles, homozygous dominant if you've got 2 dominant alleles. If you are heterozygous, so you've got 1 dominant and 1 recessive allele, that means that you will show the dominant trait because, dominant versus recessive. If you've got 1 dominant, it's going to overpower, the other allele. So in terms of notation, your dominant allele is marked with a capital letter and your recessive allele is marked with a lowercase, but they'll be the same letter. So this would be your heterozygous, this would be your homozygous dominant, and your homozygous recessive. So that's what you can kind of see there. So you would have, so capital H represents the dominant allele, which is brown eyes. The lowercase h is blue eyes. So you would have brown eyes because you're dominant, homozygous, dominant, homozygous dominant. And then this would be homozygous recessive. And as this one is heterozygous, it would be brown eyes as well, because again, you always show the dominant trait. So that's kind of when things are clear cut. You either have blue eyes or you've got brown eyes. With codominance, this is when basically you've got more than 1 dominant allele. And codominance, as the name suggests, is when they're both expressed. So a good example of this is your blood type. So you've got your ABO, blood system. So if you are an AB blood type, you obviously express both. So you've got the A allele, the B allele, or no allele or no antigen. And that's ultimately what O is, as you can see here. But if you have A and B, as we can see with these 2 green ones, you don't have blood type A or you don't have blood type B, you've got blood type AB because they're codominant. So one isn't more, powerful than the other. You express both of them. So hence the term codominance. If we think about a different side of this, it's incomplete dominance. So rather than both being expressed fully, so if you've got, the A allele and the B allele, you would express the A antigen and the B antigen. With incomplete dominance, you've got both dominant alleles, so you express a mix of both. So as you can see here, we've got the pink flower and the white flower, and they're both dominant. So rather than expressing, pink leaves and white leaves, which would be codominance because you're expressing both of the traits fully, incomplete dominance is when you've got a little bit of this mix. So you've got kind of a pale pink color that represents kind of this mix of, hot pink and white ultimately. So that's that idea of incomplete dominance. So hopefully the distinction between codominance and incomplete dominance makes sense. So the difference between a genotype and a phenotype, genotype is the actual allele that one's been given. So, you're heterozygous or homozygous dominant, homozygous recessive, and your phenotype is what is expressed. So your genotype, you could be heterozygous. A different genotype would be homozygous dominant, but both a heterozygous person and a homozygous dominant person would express the same allele. So if you had, obviously there's no person here who's homozygous dominant, but if you had two, capital B alleles, so two brown eye alleles, you would express brown eyes. And if you're heterozygous, you would express brown eyes as well. So the phenotype is the same, but the genotype is different. So hopefully that makes sense. Genotype, just think of the actual genes, the actual alleles. Phenotype, you don't care what's going on genetically. It's just what you see on the outside, basically. Okay, so your phenotype can be influenced obviously by your genes, but also by environmental factors, epigenetic factors as well. So for example, you've got someone with, blue eyes, but maybe they put in contacts. You've got someone with blonde hair, or maybe they dye it a different color. This idea that your phenotype, so the characteristic that is expressed kind of outwardly, that can obviously be influenced by the environment. And epigenetic factors as well. So epigenetic factors are basically how your genes can be altered as you live, almost. So for example, things like smoking or other things like that, these might be environmental factors. They can influence genes, and then this can be passed down throughout an individual, for example. So we think about these things. I don't want to, get into too much detail, but you can have ultimately the histone. It's kind of about which genes can be read more easily and which can't. This is when we think about epigenetics. It's kind of controlling or changing the expression of these genes. If you've got a histone, you can basically, okay, it's a bit more complex than I'll explain it, but the kind of analogy you can kind of think about it as is you are winding the DNA around the histone really, really tightly. So then it's kind of hard to open the gene and read it and express it. So that gene doesn't get expressed that much. Whereas the other influence of epigenetics can be you're sort of loosening the tightness with which that DNA is wrapped around the histone. So then it's easier to read and that gene is expressed more. So that's the sort of idea there. Okay, so monohybrid crosses. So this is when your individual alleles become really important. You might, know as Punnett squares, things like that. So it's when you are basically seeing what the offspring, what the outcome might be of two individuals with, different alleles. So here we've got a homozygous dominant and a heterozygous person, or organisms, and where you're ultimately using this square, this monohybrid cross, to see what the chances are of their offspring having certain alleles. So you can see you would get a homozygous dominant, you could get heterozygous, and you could get heterozygous as well. So I might just flick back. You can see with this one, it's obviously different. So you've got a 50% chance of the offspring being heterozygous, 50% of it being homozygous recessive. The other one here is sort of 50% the other way, 50% homozygous dominant, 50% heterozygous. And so it just gives you kind of an idea about what the offspring would express. And you can see here that there is no chance that any of their offspring would have the recessive phenotype or the recessive, express the recessive allele. So for example, if we're thinking about eye color here, if your capital H is brown eyes and your lowercase h is blue eyes, if you have a homozygous dominant person with a homozygous recessive person, heterozygous person, there's no way that their offspring will have blue eyes because there's no option here where you've got homozygous recessive. It's the same thing if homozygous dominant was to pair with homozygous recessive, because the thing is, if you've got someone that is homozygous dominant, the only allele they can give is the dominant one. And if the only allele they can give is the dominant one, there's no way that their children could have the recessive allele, if that makes sense. So this, say this is an individual, they could have 100 children, and unless it's a mutation, they won't be having someone with blue eyes because they don't have that allele. And so obviously, the offspring may get the recessive allele from the other parent, but you need 2 copies of that recessive allele to express the recessive phenotype. Okay, Hopefully that makes sense. You can see, yeah, for all 4 of these, they would express the same phenotype. So we've talked about the sex chromosomes here. Again, this is what you're looking at on your karyotype. So you've got an X and a Y for males and 2 X chromosomes for females. So in terms of how that plays into genetics, with the X chromosome, it's obviously a lot longer than the Y chromosome. So your what we call X-linked inheritance is more common because you basically inherit more genes from that X chromosome. So if you've got a certain trait that's X-linked, it's more likely that that will come from the X chromosome. Hopefully does that make sense? Yeah. You're more likely to have something that's X-linked than Y-linked just because there's a lot more space for genes, ultimately, is what I'm trying to say. Hopefully that makes sense. So we can see here, the idea is that with your X-linked trait, so let's say we've got a genetic disease that is found on a gene on the X chromosome. If you are a male, you only need 1 copy of that allele and you will express it. Whereas if you're a female, you'll need 2. So hopefully that makes sense, because if you think about, I think color blindness is a common one they use. It is X-linked. Is that right? I think that's right. Yeah. It's X-linked. The idea is that if you have this little gene for color blindness on the X chromosome, as a mother, if you have 7 sons, they will all be colorblind, if that makes sense, because you only have X chromosomes that are colorblind. And so because the son, oh my gosh, hopefully I'm explaining this, but because the son gets 1 X chromosome and the mother is giving X chromosomes that have colorblindness, the colorblindness gene, they're only getting 1 copy of that X chromosome. So hopefully that makes sense. Whereas if you are female, you'll get an X chromosome from one parent, X chromosome from the other. So if your mother is colorblind, you may get that colorblind gene. But then if your father isn't, you won't be colorblind because you don't have 2 copies of that. Hopefully that makes sense. I'm so sorry if that was the worst explanation ever. But it's just that idea that females almost have like a little backup because they've got 2 X chromosomes. Whereas if you have a genetic disease on an X chromosome and you're a male, you're going to be expressing that. Hopefully that makes sense. Okay, moving on. But yeah, a Punnett square is basically exactly the same. You just denote the little, so let's say this is like color blindness. You just denote the little thing on your X chromosome. So you can see here, this would be the colorblind one if it's the recessive allele. Okay. But then as you can see here, if this is for color blindness, we've got 1 that is not for color blindness. So you won't be expressing that. Okay, hopefully that makes sense. So looking at pedigree charts, we're not going too into detail with this, but you can look and observe traits or diseases that have been passed down and figure out if they're autosomal dominant, if they're recessive, if they're X-linked, if they're Y-linked. So we can see here, the pattern here is autosomal recessive. So we can see here, this is a good pair to look at. If you've got 2 parents who are autosomal recessive, all of their children will have the same thing. So if you've got, let's say blue eyes, blue eyes is recessive, your brown eyes are dominant. If you've got a couple with blue eyes and they're both homozygous recessive, which they would be, all their children will have blue eyes. All their children will express that trait. Again, it may not be that simple, there may be other genes controlling things, but that's the idea there, that if you have 2 parents who are homozygous recessive, the only allele you have are those 2, those recessive ones, so that's what your children are going to express. Whereas you can see here, maybe, for example, not too sure, but maybe in this case, this person may be homozygous dominant, so that's why all the children, or I should say none of the children, show the disease or the allele. And then I should mention the way to figure out if they're showing it or not is if you have a colored in box. But as you can see here, we've got heterozygous or homozygous dominant. The idea is that they're not expressing the recessive allele, but one of their children is homozygous recessive, which means someone in here, there has to be an allele somewhere in there. Both of these people are not homozygous dominant. They must be heterozygous. Because remember from our Punnett square, if one of these people were homozygous dominant, it wouldn't be possible to have a child with a recessive genotype, homozygous recessive genotype. Okay, so looking at chromosomal abnormalities, again, we've looked at what a typical karyotype should look like. So some of the syndromes that are associated with additional chromosome abnormalities. So Klinefelter syndrome is one. So here, instead of having 2X or, one X and a Y, we've got basically all three. So we've got two X's and a Y. So what the result is ultimately kind of a mix of certain traits from females and males. So this idea of things that you would see in female puberty, so kind of breast development. We've got fewer chest hairs, wide hips, for example, and kind of less of the sort of male puberty traits. So we've got like poor beard growth, poor hair growth, as I mentioned before, small testicular size, things like that. Frontal baldness is what we see in this individual, because you can see it's just traits from both, but particularly because you've got two X chromosomes, that's where these kind of traits are coming from. So Turner syndrome is when you have only one X, so you're basically missing either a second X or a Y. So this is your Turner syndrome. So you can see, again, some of the typical traits associated with this. So a very small height, so quite a bit shorter, different kind of expressions of the organs, for example, the kidneys. A web-like neck, that's a very characteristic thing of Turner syndrome, and some of the other things as well. And then Down syndrome or trisomy 21. So the name trisomy 21, it means you've got so 3 of the 21st chromosome. So when you should have 2, you've got 3 chromosomes instead. And then again, there are typical features associated with trisomy 21 as well. Okay, so the reason for these chromosomal abnormalities is because of this thing called non-disjunction. So basically your chromosomes aren't separating properly, and so you're either getting an extra chromosome or one less. So you can see here, you've got your 2 chromosomes, they're meant to separate, and then they're meant to separate chromatids again. But you can see here 2 chromatids have gone to here, and none to here. So the result is you can either fertilize and get, again, an extra copy of a chromosome, or you can fertilize this and get one missing chromosome ultimately. And then the same thing is seen here. It's just this idea of non-disjunction, and it can happen at different steps along the way, but it's this idea that the separation of chromosomes isn't happening properly. Okay, so we've got mitosis. So mitosis is this idea of your cellular division. So it's when you're growing, you're just producing more cells. And we're thinking of eukaryotic cells here. So we'll talk about different forms of division, and you often compare it to binary fission, which can be seen as a form of asexual reproduction as well. Binary fission is a lot more simple. Mitosis is regarded as being a little bit more of a complex process. And again, it's just that idea that eukaryotes are a little bit more complex than, bacteria and things like that. So here's an overview. We'll look at these steps in detail, but it's just important to know what each of the names are and what the main thing happening is. So, mitosis, we're obviously on a specific part of the cell cycle here, but it's good to know what else goes on in the cell cycle. So that's what we call interphase. And it's just when you've got stuff like, your DNA is being replicated, or your organelles are growing, the cell's growing, it's just doing its thing, hanging out, getting prepared for mitosis. So after that process, we then get to the actual cell division. So you've got prophase. So prophase is your initial step. So you've got prophase, metaphase, anaphase, and telophase. These are your 4 steps of mitosis. A way to remember this is by PMAT. So P, the first one of PMAT, is for our prophase. So we've got our condensed chromosomes So again, when the chromosomes are starting to become visible here, so we can see that they're just kind of condensing, and we're just getting ready to split, basically. So we're condensing our chromosomes, they've been replicated, and we're getting ready to split them into two. Our kind of centrioles are getting ready as well to be split. So we then move on to metaphase. So M for metaphase, you can also think about the middle. I think that's a common one to think about. So prophase, everything's just getting ready. Now with your metaphase is when they actually start to properly move. So they line up, as you can see here, sort of in the middle, and then they're just getting ready to split apart. Ultimately, your centrioles are these little structures here, and they're starting to attach to the centromeres of your chromosomes, and they're lining up at separate poles, getting ready for them, to give the chromosomes a bit of a yank. Okay, so you got your anaphase. So your anaphase, you can think of A for apart. That's how I like to do it. So M, metaphase, M for middle, A, anaphase, A for apart. So we're basically pulling our chromosomes apart. So we can see that here. So your spindle fibers that extend from your centrioles have attached to the centromere, and they basically contract, the spindle fibers pull, and when we, if you recall what we looked at before, our little sister chromatids, they get pulled apart. So we can see that here, and the idea is that because they are, part of the same chromosome, because they've been duplicated, or replicated, when you pull them apart, you're getting one copy to one side and one copy to the other. So you're not missing any genetic information. You just, made two copies of things, and now one's going to one side and one's going to the other, but they should be identical. Okay, and then we have the last part of mitosis, which is telophase. So this is when the actual separate cells start to form. So you've got your nucleus kind of reforming. Again, you've got one on one side and one on the other. And then you've got the actual entire plasma membrane that's kind of starting to fork in as well, as you can see here. And so that's that idea of telophase. The actual final separation is cytokinesis. So this idea of these cells actually dividing and splitting. This is a step that is considered sort of post-mitosis. Sometimes it can be seen, like people can call cytokinesis a stage of mitosis. Technically, it's only your PMAT. So your prophase, metaphase, anaphase, telophase, that's mitosis done. And then cytokinesis is when you have that final sort of splitting of the cell. So hopefully that makes sense. Again, the way I like to think about it is prophase, just your initial kind of getting ready. Your metaphase, your M, lining up in the middle. And then your anaphase is when you're pulling them apart. And then your cytokinesis is them being fully separated. Did I just miss telophase? I just missed telophase. Your telophase is when they're kind of being, separated. And then cytokinesis is that final sort of cleavage or cleave. Okay, so in terms of genetic variation, so this is when we start to think about meiosis and kind of looking at cell division, but then also the production of gametes. So that's when we start to get in things like prophase 1. We ultimately have it's basically the exact same steps as this, but you are doing it twice almost. So that's why you get a haploid number. So here we have basically double the amount of chromosomes that we need, and then we're separating them. So now we have the appropriate number of chromosomes that we need. But then with meiosis, you basically have another one of these sort of mitosis processes. And then so you divide those cells again, and then that's why you end up with 4 haploid cells, as opposed to, in mitosis, 2 diploid cells. Okay, so we'll look at a diagram of this in a second, but crossing over and recombination, it's basically this idea of when your homologous chromosomes, almost, I like to think of it as like holding hands and kind of exchanging genetic information. I don't know, let's say like you've got jewelry on one hand, you hold hands with your friend and you swap jewelry, something like that. It's this idea of them coming to contact and exchanging genetic information. And then independent assortment of chromosomes is just this idea of when your chromosomes ultimately get shuffled around randomly, basically. So it'll make more sense when we look at a diagram in a second, but those are some really important things to bear in mind when we think about meiosis, because the whole point of meiosis, unlike mitosis is just cell division. We just need to make more cells, we need to grow. Meiosis is when we need to create gametes to create offspring, ultimately. So the idea here of genetic variation, so what we've just discussed here, is really, really important, and ways to increase that are really important as well. So we can see an overview of meiosis here. So you've got your interphases we were discussing before, and then you go into your prophase, your metaphase, your anaphase, and your telophase. And that's ultimately very similar. You then go into your second lot of this. So your prophase, your metaphase, your anaphase, and your telophase. So the exact same thing is happening. Your metaphase is when they're lining up in the middle. Your anaphase is when they're being pulled apart. Your telophase is when they're kind of closing up, and then ultimately cytokinesis as well. The end result, though, is that you've got 4 haploid cells. So we'll have a look here. You've got your 2 homologous chromosomes. So your homologous chromosomes, again, you've got your metaphase, your anaphase, and your telophase. They separate. You then have your metaphase, your anaphase, and your telophase. And you can see that your homologous chromosomes, so you've got basically those 2 kind of copies, you're able to split them, and then they enter this cell, and then you've got a haploid number, as opposed to here where you've got your diploid number. So that's the idea there. You have, with mitosis, you have 46, you ultimately have 46 chromosomes at the start, but they look like this. So they're kind of doubled up, right? Because you've got that extra genetic information. So in the first phase of meiosis, you've got your homologous chromosomes and you split those up. But then because they've been duplicated, because they've been replicated, we're able to go through this metaphase, anaphase, telophase again. And then our 46 chromosomes become 23. So hopefully that kind of makes sense. It's just this idea of I like to kind of think about it, if you've got your homologous chromosome, so you've got a copy from mum and a copy from dad, right? Is how you can think about it. You then duplicate them. So you've kind of got 2 copies from mum, 2 copies from dad. Ultimately, you go through this process and they split. So now you've got 4 cells, 4 gametes, one of dad's copies, the other of dad's copies, one of mum's copies, the other of mum's copies. That's what you can kind of think about. But bearing in mind, I do like this diagram because if you have a look at it, these 2 chromosomes here are a little bit reshuffled. This is what we were talking about in terms of that crossing over. So you can see here, hopefully you can see, I know the diagram is a little bit small, but the chromosomes are pairing up, and you can see by the time we get here, we've got little bits of light blue on dark blue and vice versa. That's that idea of crossing over. They've kind of shaken hands and said like, okay, let me give you a little bit of my genetic information, you give me a bit of yours. And that means that in these gametes, we've got even more sort of genetic variation. So, I was talking about like, if you had one chromosome, this one's like dad's chromosomes, this one is dad's chromosome, this one is mum's, this one is mum's as well. They're not genetically identical anymore because they've shared a little bit of information. So if let's say that one of these was to be fertilized, this chromosome is not identical to the one that, your mother gave you, for example, because now it's got a little bit of stuff from dad, and then now you give that to your offspring, and it's a little bit different as well. Hopefully that makes sense. Okay. So we've talked about how they're haploid. So this is a diagram of the table that we were just looking at. So you're crossing over, this is what we've just mentioned there, the idea of when you swap a bit of genetic information. And then independent assortment is just this idea of the chromosomes that get assigned or allocated to each gamete are completely random. So here we're looking at this with one chromosome. So let's say this is chromosome 1. But remember that this process happens with 23 different pairs. So we've got 4 gametes here, and you can see that they've all got different things, that are coming from chromosome 1. The same thing will happen with chromosome 2, chromosome 3, chromosome 4, chromosome 5, and so on. They'll all have 4, gametes that come out looking like this. And, which one of these 4 gets paired up with the 4 from chromosome 2, gets paired up with the 4 from chromosome 3, and so on. That's all random. That's this idea of independent assortment. So you can see that here you've got gametes from outcome 1, gametes from outcome 2. And the idea is that within these gametes, you've got kind of completely different genetic information going on, which is why, siblings look different ultimately, because you've got different genetic information in the ovum, different genetic information in the sperm. And then obviously that is different as well when they come together. Hopefully that is all kind of clear. Please ask in the chat if any of that isn't making sense, but I hope that is kind of understandable. Okay, so then again, we're already talking about this. We're already like, okay, wow, we've got such a great genetic variation here. Obviously, the actual fertilization is even more different. This idea of you have, however many millions of sperm coming and fertilizing this egg. Again, it's different genetic information. So you've got however many sperm released. And again, this is what's going on in all of them. All of those sperm have got different assortments of, chromosomes of genetic information in them. And then they now come to this ovum and they mix with the chromosomes in there. It's just a lot of different variation, which again is why siblings can look so different and is why different people look so different. Okay, so then obviously you've got 2 haploid cells. They fuse together. You've now got diploid cells. So you've got 23 chromosomes from the sperm, 23 chromosomes from the ovum. Together, this fertilized egg, you've now got 46 chromosomes, which is exactly the number that you need. Okay, so this just shows, again, the process of meiosis to create sperm and to create ova. So something to note here is we've got our, so you can see we've got our homologous chromosomes here. So one goes into one cell, one goes into the other. You then split them at the centromere, and that's what you've got. And then you've got your genetic information within 4 sperm. So then that's 4 sperm cells. Great. With the ovum, it's a little bit different. So you've got 4, ova, let's say. 3 of them will basically degenerate. One of these 4 gametes will kind of grow and become the mature ovum that's released. These other 3 are called polar bodies. They'll basically just wither away ultimately. Okay, so this is just showing mitosis versus meiosis, and this is what what I was describing before in terms of binary fission, which is what you see in bacteria, for example. Binary fission is rapid, super quick. It's like what you think about, when you think about bacteria growing on their little agar plates. Happens super quickly because you literally just have to copy this circular chromosome and cut it in half. That's it. That's literally binary fission, very simple. Whereas mitosis, as we've talked about, is a little bit more complicated. But you start with this cell. Again, you've got your prophase, you've got your metaphase, you've got your anaphase, ultimately your telophase and your cytokinesis. So you start with a diploid number and you end with 2 diploid numbers. So these cells are identical to this first cell. With meiosis, you've got these homologous pairs. The homologous pairs are split. Then these chromosomes are split in half. Then you've got your gametes, which are haploid. So hopefully that makes sense. So this is what I mean when you've got, like, let's say that this is your copy from mum and this is your copy from dad. This is a different chromosome, your copy from mum, your copy from dad, for example, right? Whereas here you can see this may be your copy from mum, this may be your copy from dad, this is another copy from mum, this is another copy from dad, for example. So hopefully the difference between mitosis and meiosis is making some sense. And this is just another diagram that represents that. So you can see here, you split this, the sister chromatids, whereas here you split the homologous chromosomes and then the sister chromatids. Okay, Lovely. So, in terms of mutations with your chromosomes, so these are when you've got quite big mutations, because if you've got a mutation in a chromosome, that's a decent chunk of, could be a gene, right? So you could have part of it deleted, so your long chromosome becomes short. This is obviously a bit of a worry because then, that genetic information that you've lost could be important. Duplication, now you've got double the information. Inversion, you've just swapped it. So again, it's very important with genes. Everything is about that specific base sequence. So with all of these, you might think, oh, duplication isn't that bad, like as long as you've got the stuff there, or like inversion isn't that bad as long as the gene is there somewhere. But if you invert something, let's say in the middle of a gene, that specific nucleotide sequence is now messed up. The results of that could be bad as well. Insertion, this one may not be as bad. Again, it just depends if you've inserted a whole gene or like half a gene. But as long as that genetic material is kind of translocation, I should say. As long as that genetic material is kind of somewhere, it may not be as bad, let's say, compared to deletion. But that's just the idea here. So you're kind of taking part of chromosome 4 and chucking it on chromosome 20. And then translocation is just chromosome 4 and chromosome 20 swap. Okay. So looking at polyploidy, so we've talked about haploid and diploid. So again, haploid is with your gametes, diploid is with your sort of typical somatic cells. Polyploidy is when you have doubles or triples of everything. I shouldn't say doubles actually, because you should have doubles of everything, but triples or quadruples of everything. So we looked at, like Klinefelter or trisomy 21, like Down syndrome. And there's just one additional chromosome, or, with Turner's one missing chromosome. With polyploidy, every single chromosome has got 3, or every single chromosome, there's 4. So that's the idea of you've got basically duplication of the entire set. So the idea is that it may or may not be harmful depending on what organism you are. Often in plants, it's not really a bad thing. For example, I think like often strawberries are polyploidy. Yeah. So strawberries, this idea that they may be made on purpose to be polyploid because it makes them a bit bigger. And yeah, other plants, it may not be that bad. With animals, or like humans, for example, if it's sort of like everything, like every cell, it's not necessarily viable. Often it's not viable. But if it is just certain cells, for example, some specific cells in the liver, then it may not be that detrimental to health. So it just depends on what the organism is, like if it's affecting every cell in the body or if it's just affecting some cells. But yeah, generally, if you've got 3 copies of a chromosome, if you've got 4 copies of a chromosome, it's not the best thing to be having so much genetic information, because obviously it can increase things too much. Okay, so looking at different types of reproduction. So we've touched on meiosis quite a bit, so that's a component of this. But with asexual reproduction, what VCA wants you to do is to compare asexual reproduction to sexual reproduction. So looking at the advantages and disadvantages of, meiosis and forming, having 2 gametes come together, as opposed to not requiring basically another organism to reproduce. So we've talked about binary fission. We looked at that diagram. So you just replicate the chromosome and you cut the cell in half. That's binary fission, so a very quick process. Budding is kind of what we see here often in Hydra, for example. So we can see that it's literally, as the name sounds, they just start to grow this little bud, and eventually it grows and becomes an entirely separate organism. Vegetative propagation is literally like when you're gardening, and when you plant something. So, you can take a stem or a root off another plant and put it in soil and it'll grow. This idea of vegetative propagation as a form of asexual reproduction. So these cells are able to basically grow on their own. And then spore formation, which is what we're kind of seeing here. So you've got your plant and then your spores are basically released in the air and they travel throughout the air and then they can land somewhere else, often in a moist environment, and then grow from there. So all of these examples involve asexual reproduction. So they come just from one organism itself. What that means, though, is that there's none of what we've talked about before in terms of the genetic variation. So you don't have this additional sort of source of genes or source of alleles as you do in sexual reproduction. And that's a really big consequence, or a really big negative of asexual reproduction, is that they are ultimately clones of each other. So like this little hydra, this is genetically identical to this. And so that's not the best thing, because of this idea, if there's no genetic diversity, the species can ultimately be wiped out. And we'll talk about that when we get to biodiversity. But that's the idea there. That's a really big negative, is this idea that you have less genetic diversity. A big positive of asexual reproduction is this idea that you can replicate quite quickly. So with binary fission, for example, if, I don't know, let's say that the bacteria is running low on numbers, or any other organism that uses binary fission, the idea is that they can replicate really, really quickly to get a large amount of offspring or of other organisms in a really short amount of time, compared to sexual reproduction, which often takes a little bit longer. So that's the idea there. Okay, so with reproductive cloning, so you ultimately create a clone, as the name suggests. So you can kind of think of it as like, it's like sexual reproduction, but you don't need the fertilization. Hopefully that sort of makes sense. So you've got your somatic body. So this could be a cell from anywhere. So let's say I wanted to clone myself. So I would take a somatic cell. So let's say maybe I took it from my arm, my stomach, wherever. So I take this body cell, and it's got obviously all my genetic information in the nucleus. So I take my nucleus out of this cell. Let's say it's from my skin. I take a skin cell. I take my nucleus out, and that's got all my DNA in it. At the same time, I take this egg cell. So it could be my egg cell, could be anyone else's egg cell. So we take this ovum and we remove their nucleus. So we remove the foreign, genetic information from the other person or the other organism. So now we've just got basically an empty ovum. And we then put my nucleus that came from my skin cell into this ovum, remembering that the ovum is able to kind of generate and grow rapidly. So we put my nucleus into that egg, and the egg does, what the egg should do. It just grows. It makes a lot more cells because it recognizes that we've got 46 chromosomes in here. So let's get growing, and that's what it does. And ultimately it grows into another organism, and then I clone myself. Obviously that hasn't really come to life yet, but I think with animals it's a little bit more common. Has been done before. So like Dolly the sheep is a big thing because it was the first animal to be cloned, I believe. So that's exactly what they did. So they got Dolly's mum. They got Dolly's mum's nucleus. They got an egg cell from this random sheep. They put Dolly's mum's, Dolly's mum's, Dolly's mum's nucleus into this egg cell, and they then inserted this egg cell into another sheep, and then that sheep gave birth to Dolly. So hopefully that makes sense, and that's the idea here of reproductive cloning. So we take this cell, and so I hope it makes sense what I was saying at the start in terms of it's like sexual reproduction but without the actual fertilization. So you've ultimately gotten, instead of getting 23 chromosomes from one, 23 chromosomes from the other, putting them together and then letting it grow, you just get 46 chromosomes from one, put it in the ovum, and then put the ovum in the surrogate, and then that egg continues to grow. And then you've got a clone because obviously there's no genetic variation. So hopefully that makes sense. Then Dolly the sheep was a clone of the other sheep. There are obviously negatives associated with this, lots of ethical things, as you can imagine. So that's probably a really important question that might come up. With the new bio study design, so basically bio got a new study design in 2022. So you guys are the second year 1-2 cohort to be using this study design. A big change in that study design was a lot more emphasis on bioethics. So going into 3-4 next year, you can expect to see a lot more bioethics questions. So this is a really good example of a spot to discuss bioethics. So I'm sure your teachers might bring it up when discussing this. But you want to be thinking about things in terms of ethics, ethics of cloning, of removing this egg, things like that. And you can think about it in different categories. So kind of like moral ethics, or perhaps, economically, consequences of that. The health of the actual clone, the health of the surrogate. Because on that note, I think with Jolly the sheep, their idea of cloning is that I think you talk about telomeres in 1-2. So telomeres are like little caps on the end of your chromosomes, and they basically give the chromosomes an age, if that makes sense. So they, like, the older you are, I want to say shorter your telomeres are, either shorter or longer, but that's the idea. They're like a mark of age, basically. So when you get a somatic cell, so let's say, I get a somatic cell from myself, I take it out and I make a clone of myself, right? So I have it in a surrogate. Obviously it starts as a baby. But the idea is that the telomeres in the genetic, in my genes, in my DNA that we've now used in this clone, are short because I'm older, right? So then when you put it in this baby, or let's say like Dolly the sheep was obviously, a lamb. The idea is that her telomeres were shorter. So I think she died pretty soon after, I want to say. So that's the idea there. Like, there's a lot to kind of think about and consider. It's not really just like, oh, you can just take this and chuck it in and then have a clone. Okay, so in terms of therapeutic cloning, this is obviously different to reproductive cloning. Reproductive, you're thinking about reproducing an organism, as in like giving rise to something else. Whereas therapeutic cloning is when you're more so just looking for the actual cells. So you have this clone ultimately, and then you can use these cells for tissue culture. So for example, you might use stem cells or things like that when, perhaps like in leukemia or in different other contexts with diseases, when you require other tissues or tissue regeneration, something like that. You can think about that as cloning, because you're using this genetic information to make other cells, perhaps to replace cells that have been depleted somewhere else. So reproductive cloning and therapeutic cloning are two different things, so don't get them confused. All right, so we've talked about some advantages and disadvantages already. So if we think about it in plants, it might be a little bit more ethical than in animals, but you can see things like increasing yield, disease resistance, because obviously you can just get a plant that's really good, that's got all these perfect traits. You just take their genetic information and you clone them, and you can do this 1 billion times, and now you've got this amazing, crop of really great, strong plants. So you can see that a lot of these advantages are very helpful, again, in the context of plants. Disadvantages, so a really big thing, like what we've talked about with asexual reproduction, is lack of genetic diversity. So again, things can be wiped out. So if you've got, this amazing crop, this amazing field of, plants that are genetically identical, they're great, but they're genetically identical. If a disease comes through, if one's susceptible, all of them are susceptible because they're genetically identical. So that's something to really be aware of. And this idea of deleting alleles from the gene pool. So, you've got other alleles such as poor yield, maybe, late fruiting, I don't know, disease susceptibility. Obviously, they're not alleles that you want, but the idea of completely eradicating them from the gene pool is, again, a disadvantage, because that idea of genetic diversity, and just creating things that are really genetically similar is not advantageous in the long term, ultimately. Okay, hopefully that is all making sense. Please let me know if there's any questions in the chat. But with biodiversity, we're thinking about that now, kind of going on from that. So this is a really big one where you have to know all your definitions. There's a lot of separate terms, I want to say. So knowing having your glossary and kind of keeping that up to date is important for something like this. I would say biodiversity is a little bit more straightforward than some of the other stuff we've looked at, particularly, your complex processes of meiosis, mitosis, and your chromosomes and all that. But don't let that kind of lull you into a false sense of security, because again, the main thing with bio is being able to answer your questions properly. And with biodiversity, it's a lot of application questions. So it's a lot of, here's a nice chunk of article, here's a lot of reading to do. Now tell me about this, and apply this in an unfamiliar setting. And that's something that practice questions are really, really important for, which I'll talk a little bit later. Okay, so in terms of your definition here, closely related organisms that are similar to each other and usually capable of producing fertile offspring. Next year in 3-4, you talk about the definition of species again in this context of when one species becomes two, when animals become different. So this idea of producing fertile offspring is a really important definition, because that's a way of figuring out if species are different. So I think if you think about a mule, which is a donkey and a horse, I believe. So a donkey and a horse, they can mate and they can create a mule. And that's great. A mule can be, a mule is fine. It can be used for different things, or it's fine. So it's viable offspring because it can live. It doesn't die instantly at birth. But the reason why horses and donkeys are two different species, I think it's horse and donkey, are two different species, is because mules are not fertile. So a mule cannot mate with another mule and have offspring, ultimately. So that's the idea of why they're two different species. So it's a really important definition to understand. Okay, so again, as the name suggests, biodiversity, it's just the diversity, the variety of organisms on Earth. So biodiversity, again, is something that comes up next year when you look at this thing, to do with speciation and all this sort of stuff. But the idea here is that the more biodiverse something is, the better. The more genetic variation, the better, as we've discussed previously. So the way for things to be biodiverse is a large population. If you have a small population, you're not going to be as genetically diverse because you have less members ultimately that have less different, characteristics. At the same time, you can have a large population and they can all be genetically similar, which isn't that good. But generally, the larger the population is, the more alleles you've got in there, the more they're mixed around, the more they're different. That's beneficial. And the main thing from this is that the species is more likely to survive. Because again, the large population, you've got large numbers there. So if you got something that's going to wipe a lot of people or organisms out, you're kind of safe. But at the same time, if you've got a disease and it targets this sort of, not targets a specific gene, but if you have certain members of the population that are susceptible, if everyone is pretty different in that group, then it means that some people will be susceptible to this disease, but other organisms in that population that are genetically quite different, they may be immune or not susceptible, is the idea there. So they're less likely to go extinct. So that's a really, really big thing with biodiversity. So in all your short answer questions about biodiversity, that's what you're going to want to be talking about. The idea of survival, the idea of going extinct. Really, really important. Include that in all your short answer questions. And the last point, so being genetically diverse allows the species to adapt to changing circumstances. So again, this idea, if you've got a disease and it's coming through, we'll talk about natural selection in a second, but this idea of you've got sort of survival of the fittest. So if you've got, a large population, organisms that all are capable of different things that are all suited to different things a little bit more than others. That means that if a disease comes through, if the environment rapidly changes, if a new predator comes, all these sorts of things, because individuals are diverse, you've kind of got at least parts of the population that will be able to survive in whatever circumstances, if that makes sense. Yeah. Okay, so strategies for maintaining biodiversity. I'm sure you're all aware of this idea of, declining biodiversity and ways to improve and maintain that. So this idea of tackling climate change, obviously with climate change, you're seeing, decreased habitats, you're seeing destruction of environments, and obviously that negative consequence on plants and animals. This idea of overexploitation, decreasing pollution. So this idea of human sort of habits almost. So, industrialization and creating housing and things like that, and obviously the use of transport, all, important and beneficial to humans. But the idea is that biodiversity is suffering as a cause of that. So kind of being aware of things like that, conserving species, conserving habitats, and then management of invasive pests. So if you bring in an introduced species, and this idea of it kind of wiping out certain organisms, because of this idea, it's basically like a disease. Because if you think, like, if you are bringing in a disease that nobody has ever had before, nobody has ever been exposed to it, you don't have immunity to it, and you're going to get really sick, and everyone's probably going to suffer, right? They're all going to be susceptible to it because they don't have any previous immunity. It's sort of the same thing with these invasive pests. If you bring in these foreign animals to different habitats, to different ecosystems, these animals haven't seen this before, and they're likely to be quite vulnerable to changes that those pests create in that environment. Okay, so adaptations, this is an important thing as well. Again, it's very application-y. VCAR doesn't want you to look at a billion different species of organisms and remember all of their adaptations. It's this idea of you're probably going to get an animal, you'll get a scenario, you'll get a description of a certain characteristic. They have maybe a certain description of a characteristic that a different, organism has. And then why one's better suited to the environment. That's sort of the common question that you might come across. You should know the different categories as well, if it's a behavioral adaptation, if it's a physiological adaptation, if it's a structural adaptation, and how, again, it benefits the organism, how it allows them to survive. So behavioral, we're often thinking about things, to do with mating, like a certain call or a certain, dance. Sometimes you can see with birds and like tropical birds and things like that. These are behavioral adaptations because they increase the likelihood of mating, and obviously that increases the chance of offspring survival. Physiology, so kind of the idea of the almost the genetic makeup of a certain organism. So for example, if you have a longer, what we call like a loop of Henle, so in your kidneys absorb, they do a lot of things, but they absorb water, right? So if you're in a really dry environment, if you've got a longer loop of Henle, you can absorb a lot more water and you can stay more hydrated compared to another animal that may have a shorter loop of Henle. They won't be able to stay as hydrated in this dry environment. So that's kind of physiological example. And then structural, maybe you're thinking about, size or shape of wings, or, color of fur or something like that. So adaptations develop due to natural selection. So this idea that the whole sort of species or this whole group of organisms can change over time is this idea, because again, it's survival of the fittest. So if you've got some organism that has a long loop of Henle and another that has a short one, the short one is more likely to die because they're going to be less hydrated, whereas the one with the long loop of Henle is more likely to survive, and more survival time equals more breeding time equals more offspring. So then over time, you're going to start to see these organisms generally have longer loop of Henles, and this idea of it being an adaptation that develops over time. Because again, survival of the fittest is this idea that if you've got something that's more suited to this environment, it's going to show up more because they're the organisms that are going to be living longer and breeding longer. Okay, so hopefully that makes sense. Again, these are just some other specific adaptations for this animal, but you would want to be thinking about, behavioral, are they physiological, are they structural? Okay, so here's an example of natural selection. I've kind of mentioned it with this idea of adaptations previously. This is the classic example that comes up everywhere. I'm sure you've heard of it before, but this idea of peppered moths in England. So it's important to understand that natural selection is separate to evolution. You cover evolution in 3-4, Bio 3-4 next year. Natural selection is different to that. They are intertwined and they can contribute to this process, but it's a little bit different. So just be aware of that. So going on with this example of peppered moths. So before the Industrial Revolution, you have this picture here, and this is kind of post-Industrial Revolution. So the bark of the tree is white. So you can see the moths here. The moths here that are white can camouflage more easily. So this main predator, this bird, you're looking against a white background, you're more likely to see these black moths, and that's what you're going to eat at first. Therefore, if we think about natural selection, over time, you're going to see more white moths than black moths because these white moths are kind of, they've got an adaptation. They're adapted to live in this environment because they're camouflaging better compared to these black moths, which stand out. So that's the idea. Before the Industrial Revolution, if you think again about survival of the fittest, the white moths were the fittest because they camouflaged the best, and the black moths were just being eaten left, right, and center. So during the Industrial Revolution, after the Industrial Revolution, you've got all of this ash, smog, smoke going around, and it's making the trees darker. So now you've got what we see here, these really dark trees. So now it's reversed, right? The black moths are camouflaging in really nicely. The birds struggle to see them. Whereas these white moths, which used to be at an advantage, they are sticking out, and they're really obvious to see. So these birds are obviously going to eat them all up, and their numbers are going to dwindle, while the black moths, their numbers are going to increase. It's what we've just talked about before with that dehydration example. The one that is not camouflage, or that's not suiting its environment as well, so the white moths, they're going to die because they're going to be eaten. And that means they've got less time to produce offspring. Whereas the black moths, they're camouflaging, they're hiding really well. They've got more time to produce offspring. And what that means is they've got more time to produce offspring that look like them. So they're producing more black moths. Whereas the white moths, they've got less time to produce more white moths. So that's the idea there. You can see how ultimately over time, you're going to get more and more black moths. So that's what you can kind of see here. So as the trees became darker, the population of moths became darker. And that just carries on for generations and generations. And then over time, the white moths are really kind of gone. Their numbers dwindle. They're in really low numbers because, again, thinking about adaptations, they're not suited for their environment. So that's where you can kind of understand the name as well. So natural selection is this idea of you have this sort of pressure in the environment, this almost selector in the environment, which is in this case your predator. It might be a disease, it might be the weather, it could be a lot of different things, but you have this environmental pressure and it forms the basis of who survive and who don't. So this predator is going to eat those who are less suited to their environment is basically the idea there. Natural selection questions. Natural selection is on the syllabus again in 3-4. So this is a really important example of something to be on top of. Typically in 3-4, the questions will be like 3 or 4 marks on natural selection. So I always tell my 3-4 students to plan their answers on natural selection. This is something as well, again, I'll talk about it at the end, but planning your short answers can really help you out sometimes. And when you've got a 4-mark natural selection question, this can be a really, really important example of doing that. So you want to discuss this idea of there being variation. That's your first step. So the first thing you would talk about is within the environment, and again, this goes for peppered moths, this goes for any natural selection question that you get, there's variation. And VCAR will be very obvious in the way that two organisms are different. So here you would say there is naturally variation. So some moths are just naturally darker colored, some moths are naturally lighter colored. And that's just a variation that exists. The second thing you talk about is this idea of this environmental pressure. So you have your predator and this idea of camouflaging in the background. So you've got your black moths, which are camouflaging more. They are less noticeable to the predator. The predator is more keen to eat these white moths because they're more visible. This results in the black moths having more time to survive, more likely to survive, and more time to reproduce, more time to pass their alleles. This is something that you say as well, like they pass their alleles or pass their traits onto their offspring. So they survive, they reproduce, and they pass down their alleles. And that means that over a few generations, the population starts to look more like them. You get less white moths and more black moths. And then basically that's the idea of natural selection. So the whole idea is that over time, this allele becomes very prominent in the population. So I think that's there's 3 or 4 points to remember, and that's ultimately to get your 3 or 4 marks. But a big one is always that first one, talking about variation. So that's an important thing to understand. Okay, so looking at relationships, you've got relationships between different organisms. So this idea of your ammentalism, your mutualism, etc. So some examples of this. So cow steps in mud, which kills a group of insects and plants in it. So ammentalism is this idea of when you've got one group that is ultimately being harmed, and then the other that doesn't really have an effect. In terms of commensalism, this idea of some birds living in the hollows of trees. So this idea of your protection offered there to the birds. You've got one benefiting and the other not having an effect. In terms of your mutualism, both benefiting. So bacteria in your human stomach feeds off nutrients that the human eats. So again, you're both sort of benefiting from that. Leeches suck blood out of hosts is your parasitism. So obviously parasite, you've got one is benefiting and one is harming. And your predation is basically predatory. So you just eat the other organism really. Yeah, but hopefully that makes sense. The ones that come up the most, I'd say, is like predation and your mutualism. Ultimately, that idea of both organisms kind of benefiting, so mutually. So you can see like bacteria feed off nutrients and they're favorable to humans as well. And then some of the others are really just the other party just doesn't get hurt. Either one benefits and the other doesn't care, or one dies and the other one doesn't care, is what you can kind of think about there. Okay, this is your little food web that's going on here. So you can see that you've got it's like a food chain or food pyramid, but a little bit more matched up. So you can see you have your like your producers and your consumers basically. And it's just important, again, it depends on your teacher, it depends on your SACs, but often you're asked to interpret them as opposed to drawing them, but you may be asked to draw them as well. So in terms of your arrows, it's the idea that you're pointing towards the organism that's eating the other. So your grains are eaten by your birds, which are eaten by your foxes eat your rabbits, which eat carrots, which eat grass, etc. So it's just important to understand the way that the arrow goes. But yeah, pretty self-explanatory. In terms of your energy pyramid, so we just talked about your producers and your consumers. So you've got your kind of apex predators at the top and your primary producers at the bottom. Your primary producers are, your plants, your things that are going through photosynthesis, so absorbing the sun's energy, and they're converting that into their own stored energy. every time that something consumes something else, 90% of that energy is lost. So your consumer here, so you've got your rabbit that eats your carrots, they're getting 10% of that energy. Your foxes are getting 10% of the rabbit's energy, which is 1%. Oh, is that right? Yes, 1% of the carrot's energy. Hopefully that sort of makes sense. But it's this idea that as you go up, you lose a lot of energy going up that sort of food pyramid. Yeah, hopefully that makes sense. In terms of keystone species, so keystone species are, again, as the name suggests, they're very, very important, and any fluctuation in their number will affect the kind of entire food web, or will affect the other organisms in that ecosystem a lot. So often we're thinking of predators here. The idea is that they have a really big influence, but there may be only a select few of them. So if you have a predator and this predator just sort of dies, you will get a big change in the ecosystem. So let's say that like foxes die, maybe, I don't know. Let's say that foxes die. You've now got a huge number of birds. You've got a huge number of rabbits because they're not being eaten by anything. Now your carrots are dwindling, your grasses are dwindling, your grains are dwindling, that sort of thing. Your grasshoppers are going to dwindle a lot as well. Your owls will suffer as well. So it's this idea of foxes are gone. Now there's heaps of birds. Now there's a small number of grasshoppers because the birds are eating them all. Now the owls are suffering because of that. Now there's going to be a smaller number of mice, and so on and so on. So that's the idea of why they're keystone species, because a fluctuation in their number will completely, or will cause a really large fluctuation and a large change in that ecosystem. And so it's that idea of other organisms being dependent on their numbers. Okay, so kind of going on from that, what impacts population? So you've got predation, obviously, competition. So again, if you're introducing these invasive pests, other organisms that are competing for the same resources, that's going to cause a fluctuation as well. Disease, chance environmental events, obviously the number of births. So with predation, we're thinking about like the number of deaths, births as well, in terms of migration and resources. So these are all very intuitive as to how they would impact populations, but it's important that you can, again, apply. So if you're given any sort of scenario, being able to understand, oh, okay, they're talking about migration, or okay, they're talking about competition. Now I'm going to write about this and mention how it affects a population. Okay, so a little practice question just to finish off our content. Unlike many domesticated animal species, sheep have a diverse gene pool. The merino breed of sheep is particularly diverse. This means that the merino breed of sheep what? So again, this is an example of sometimes distracting or unnecessary information. Sometimes you'll come across questions and they might look, I'm not saying that this looks hard, but you'll see foreign information and you might go into a bit of panic. So you might see something like the merino breed of sheep and think like, oh my gosh, I've never heard of this before. I haven't done any research on this breed of sheep. Or, you might get a bacteria you've never heard the name of. You might get a long little article about something that you've never heard of before. It's not always necessary. Like, for example, with this, the merino breed of sheep is particularly diverse. This means that the merino breed of sheep, blah, 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 blah. If you break this question down, it's literally just asking you what is something that is true of diverse populations. So try not to get too caught up on information that, seems foreign to you. Obviously it's different, if you get a big chunk of information, you need to comprehend it and understand it, but you don't need to know every single thing. Often VCA will use things, like this, just as a bit of added context, or even to, trick you or mess you up a little bit. But if you just break the question down, it'll be something that you know. You'll never get a question, or at least your teachers shouldn't put a question on your SAC or your assessments that require knowledge that, is too specific or that you haven't been taught. Okay, so has been through a genetic bottleneck. So no, I know we haven't talked about bottlenecks and stuff like that today, but if something is diverse, a genetic bottleneck is basically like a decrease in genetic diversity, so that wouldn't really make sense. Will be very susceptible to infectious disease. No, in fact, it would be the opposite if you're particularly diverse. Will show a high frequency of genetic disorders. Again, no, because if you're particularly diverse, genetic disorders that certain individuals have shouldn't be that prominent in the population. For example, there are populations where if the gene pool is small, or for example, they may migrate and start their own population. So if you have, let's say we've got this nice big population of 100 people, 5 people leave and go to this other little area and they start, making their own offspring and they have their own little population that's separated. Let's say that 3 of them had a certain, characteristic. You could say like they had the same colored hair, or in this case, let's say that they had a genetic disease. Obviously, if they are basically the core of the alleles in that gene pool, as they have children, all of those children are going to have the same genetic disease they had. It's kind of what we were talking about before in terms of, if you've got homozygous recessive parents, they're going to have homozygous recessive children. It's sort of that idea there. So that's where you'd see a high frequency of genetic disorders in those sort of scenarios where you've got a really limited gene pool because it may have started from only a few people. So then obviously that leaves the last one, which is D. Has a good chance of surviving environmental change, which again is true because of the things we talked about. They're less susceptible to infectious disease. You've got different adaptations that can be suited to different changing environments ultimately.